In exercise 16, you'll be doing respiratory cultures. The upper respiratory tract has normal microbiota in the mouth and the throat and the upper respiratory area, including various Streptococci, Micrococci, Staphylococci, Corynebacterium, Haemophilus, and Neisseria. Pathogens that may cause infections include Streptococcus pyogenes, Streptococcus pneumoniae, Corynebacterium diphtheriae, Haemophilus influenzae, and Neisseria meningitidis. Culture and biochemical identification is essential for diagnosis. For example, you see that Corynebacterium xerosis is a normal organism that's found in the upper respiratory tract, but Corynebacterium diphtheriae is a pathogen. They look alike on a gram stain, so biochemical identification is essential for diagnosis. This lab is going to focus on the gram-positive cocci of medical importance and on how to differentiate them from one another. The first test, once you have established that you have a gram-positive coccus isolate, is the Catalase test. The Catalase test will differentiate between the Staphylococci and Micrococci and the Streptococci. The procedure involves putting a drop of hydrogen peroxide on a clean slide. This is simply drugstore hydrogen peroxide. You'll take a sample of the unknown using a loop and simply touch it to the drop. Vigorous bubbling indicates the presence of catalase, which breaks down hydrogen peroxide to water and oxygen gas. Never use a sample grown on blood agar for this procedure. Red blood cells naturally contain catalase and you'll get a false positive result. Catalase test results show that Staphylococcus and Micrococcus are positive. Bubbling will be observed after touching the colony to the hydrogen peroxide. Streptococcus species do not make the enzyme catalase, and therefore you will not see any bubbling when the Streptococcus organism is touched to the hydrogen peroxide. Once you have established that you have a Staphylococcus or Micrococcus, it's important now to differentiate between the two. A gram stain will be useful, but if it's not done, observation of pigment in the colony can provide a clue. Staphylococci in the gram stain are seen as clusters, whereas Micrococci in the gram stain are seen as tetrads. If we look at the colonies, Staphylococci on tryptic soy agar are white, whereas Staphylococcus aureus will be golden. This is more of a buff color, not a highly pigmented color. Micrococci, on the other hand, are highly pigmented. Micrococcus luteus is a lovely lemon yellow color, and Micrococcus roseus is a beautiful rose color. The Micrococci are normal inhabitants of the upper respiratory tract and the skin, and they are not considered to be pathogens. Staphylococcus aureus is the primary pathogen and now must be differentiated from other staphylococci such as Staphylococcus epidermidis and Staphylococcus saprophyticus. The coagulase test will be used to differentiate the coagulase negative Staphylococci from Staphylococcus aureus. There are two ways to perform the test. The first is the one that we will do in lab, which is called the slide test. For the slide test, we will place a drop of plasma on the slide. Plasma comes from rabbits, although you could use human plasma, and then you rub a sample of the organism into the drop. You vigorously rub it in there, trying to emulsify it to get it to dissolve. If it will not dissolve and you see clumping, this indicates a positive test. Staphylococcus aureus is coagulase positive. Staphylococcus epidermidis and Staphylococcus saprophyticus are coagulase negative. Here you see the negative result is a creamy background. This is Staphylococcus epidermidis or saprophyticus, and you see the nice chunks and clumps in the positive test. That's Staphylococcus aureus. Staph aureus is also golden in color, and you can see that here. The other way to perform this test is called the tube test. Here the organism is inoculated into 1 ml of plasma, and it's the same plasma that you use for the slide test. It's placed in a small tube and incubated for 1 hour at 37 degrees Celsius. After 1 hour, Staphylococcus aureus will clot the plasma, whereas Staph saprophyticus or Staph epidermidis will not clot the plasma. It will still be liquid. The tube test is more reliable than the slide test, but the slide test takes less time and less plasma. Now it's necessary to differentiate the coagulase negative staphylococci, Staphylococcus epidermidis, from Staph saprophyticus. The test that we do for this is called the novobiocin test. Novobiocin is an antibiotic, and it is sold in a disc. The disc is placed on a heavy inoculum of the organism in question and incubated for 24 hours at 37 degrees Celsius. After incubation, it is observed, and you'll see a large zone of inhibition around the novobiocin disc for Staphylococcus epidermidis, whereas Staphylococcus saprophyticus is resistant to novobiocin. It's important to differentiate these two organisms, especially in samples from young females. Young females typically will get a Staph saprophyticus urinary tract infection if they are sexually active and contract this as a sexually transmitted infection. Now we have to differentiate the Streptococci. Remember, the Streptococci are catalase negative. Streptococcus ID is going to begin with inoculation onto blood agar. We use sheep blood agar in the lab, and after incubation we are going to observe hemolysis. Hemolysis results from lysing of the red blood cells, and there are 3 types. The first is beta hemolysis. Beta is much like a capital B when we write it, and this is the complete lysis of the red blood cells in the agar. The red blood cells are popped open, the hemoglobin leaves, it clears the agar, and when you hold this up to the light, you can actually read right through it. So this clear area is called beta hemolysis. Alpha hemolysis is partial lysing. The cells leak. So when the red blood cells leak, they leak hemoglobin. Hemoglobin gets reduced to methemoglobin, which results in a greenish cast surrounding the colony. It's sort of an army green type of a color, and this is known as alpha hemolysis. Sort of looks like a fish swimming to the left. Gamma hemolysis, looks like a fish swimming down, the Greek symbol, is somewhat of a misnomer because it means no hemolysis. So when you look at this plate, here you can see green for alpha, clearing for beta, and no change for gamma hemolysis. Knowing these hemolysis patterns, then, we can begin to figure out which Streptococcus we are dealing with. The beta hemolytic Streptococci are all not pathogenic, but the important pathogen in this group is Streptococcus pyogenes. We have to differentiate Streptococcus pyogenes from other beta hemolytic Streptococci that are non-pathogenic. To differentiate them, we perform a bacitracin test. Bacitracin is an antibiotic, and it is impregnated into a disc. The disc is typically marked with the letter A. It's placed on a heavy inoculum, perhaps on a T-streak of the organism, and then it is incubated. Following incubation, a zone of inhibition is presumptively positive for Streptococcus pyogenes. Differentiating the alpha and gamma hemolytic streps is done by looking first at the blood agar. Most of the Streptococci in the oral cavity are alpha hemolytic. Streptococcus pneumoniae, a frank pathogen, has to be differentiated from these normal alpha hemolytic Streptococci that might be isolated from the throat. The test that we use for this is called the optochin test. Optochin is a chemical, ethyl hydrocuprin hydrochloric acid, and it is impregnated into a disc. The disc will be marked with the letter P or OP. The disc is placed onto an inoculum of the organism, And Streptococcus pneumoniae will lyse, this chemical will lyse Streptococcus pneumoniae, producing a zone of inhibition after incubation. So you can see that Streptococcus mitis, which is a common oral streptococcus, is resistant to optogen, whereas Streptococcus pneumoniae shows this zone of inhibition, telling us that it is susceptible to optogen. We also have to differentiate Enterococcus faecalis. Enterococcus faecalis is a serious pathogen. It typically is gamma hemolytic, although it may be alpha hemolytic. Enterococcus faecalis is resistant to optogen. You will be performing a nasal screen for methicillin-resistant Staphylococcus aureus during lab. Many clinical facilities are swabbing the nares upon admission to control MRSA. You will swab your nares and T-streak the specimen on mannitol salt agar. Back in exercise 8 with the food samples, you learned about mannitol salt agar. Mannitol salt agar is differentially selective for the Staphylococci. You then will place an oxacillin disc on the primary area of the T-streak. Methicillin is no longer available, so we use oxacillin in its place. Following incubation for 24 hours at 37 degrees, you will observe for MRSA. Yellow colonies on the mannitol salt agar will be Staphylococcus aureus, and a lack of a zone of inhibition around the ox disc will indicate that the organism is resistant to oxacillin. In lab, you will also be collecting a throat culture from your lab partner. Throat cultures are collected with transport swabs, and you can see there are a variety of different transport swabs in this image. It's very important that you know properly how to use these transport swabs, because if they're not used properly, they will be rejected by the lab and you'll have to collect another specimen. These transport swabs contain a transport medium, such as Amy's or Stewart's medium, that are called minimal media. They keep the organism alive, but they don't promote multiplication. That's important because we don't want the organism to multiply in the hour or two, perhaps before it is cultured in the laboratory, because we would have a false picture of what was going on at the time of collection. There are many different brands, so be sure you follow directions. If you don't know how to use one in your job, make sure you ask someone to show you. Sputum cultures are collected in sterile containers, and here are some examples. It's very important that you give clear instructions to the patient. Sputum is brought from the lungs. It is not saliva, nor is it postnasal discharge. Never ever tell the patient to spit in the cup. Spit is saliva. You have to tell the patient that this substance should come from the lungs. Always wear gloves when handling the transport container that has a specimen in it. You never know if the patient has missed when they brought up the sputum and delivered it into the container. A throat culture is performed by first getting your ducks in a row. Have everything right in front of you that you need and work quickly. When you are dealing with a patient, you will be wearing gloves and a mask. In lab, you'll be wearing gloves, but not necessarily a mask, unless your lab partner has a respiratory infection. You are going to ask the patients to say ah, and this is going to lift the uvula, the little punching bag, in the back of the throat. You'll quickly depress the tongue, but don't put the depressor so far back that you gag the patient. Remove the swab from the container and quickly swab the back of the throat. Swab also the tonsils, but do not touch the teeth, and also do not touch the tongue. Gagging is expected. Move quickly. This is uncomfortable for the patient, so you want to get in and out very quickly. But be prepared. Gagging often induces the patient to vomit. If the swab is being done to rule out strep throat, and a rapid strep test will be done immediately, you may want to collect 2 swabs simultaneously. There are even transport swabs, transport containers that have 2 swabs stuck together so that you can do this. That's beneficial because if the rapid strep test is negative, oftentimes the doctor will want the specimen sent to the hospital laboratory for a culture. So if the test is negative, you already have the second swab for a throat culture, and you don't have to go back into that little kid who has the sore throat who wants to bite you when you take a throat culture. Carefully insert the swab into the transport device and activate the device if necessary. Label it and transport it to the lab. Some facilities will require that you refrigerate the specimen if it is not cultured within an hour. Some laboratories do not want throat cultures refrigerated because Streptococcus pyogenes tends to be rather sensitive to cold temperatures. So definitely check with the procedure manual where you are working. A sputum culture is performed to diagnose lower respiratory infections, such as tuberculosis and pneumonia. It is essential that the proper specimen is collected, and once the specimen reaches the lab, we'll be sure to make sure it's the right specimen. It will be stained to ensure it is sputum and not saliva. Sputum has many white blood cells and few epithelial cells, whereas saliva has many epithelial cells and few white blood cells. A culture will then be performed using various differential and selective media to try to elucidate the pathogen that might be in the specimen. In lab, you're going to be given an unknown. You will identify this unknown for a grade. The unknown is going to be a gram-positive isolate. You will create a lawn on each side of a TSA blood auger split plate. You can see the split plate here. Half of the plate is poured with blood auger, the other half is poured with tryptic soy auger. After you inoculate your unknown on both sides, you're going to apply a novobiocin disc to the TSA side, and to the blood side you are going to apply an optogen disc and a bacitracin disc. You'll incubate this until the next class period, upon which you are going to use the techniques that you learned in this lab to identify the isolate. See you in lab.